Friends, welcome in the world of pharmacy. I am Professor Ramdas Darade. Today we will discuss antidiabetic agents. Antidiabetic agents, these are the drugs or substances used in treatment of diabetes mellitus, which is caused due to deficiency of pancreatic hormone insulin. So, antidiabetic agents, before discussing this topic, you must know what is definition of diabetes mellitus. So diabetes mellitus, it is a metabolic disorder characterized by hyperglycemia, means increase in glucose level inside the blood. Then glycosuria, means presence of glucose inside the urine. Then hyperlipemia, means increase in lipid concentration inside the blood or plasma. Then negative nitrogen balance, means there is an increase in excretion of nitrogen from the body as compared to intake of nitrogen. And sometimes it is characterized by ketonemia. Ketonemia means presence of ketone bodies inside the plasma. So diabetes mellitus, it is basically it is a metabolic disorder. It is autoimmune disorder. It is caused due to deficiency of pancreatic hormone insulin, and it is characterized by hyperglycemia, glycosuria, hyperlipemia, negative nitrogen balance, and sometimes ketonemia. Types of diabetes mellitus. There are two major types of diabetes mellitus. Type 1 diabetes mellitus. Type 1, it is known as insulin dependent diabetes mellitus. It is also known as juvenile onset diabetes mellitus. It is known as juvenile onset because this diabetes, type 1 diabetes mellitus, occurs at the early age. Therefore, it is known as juvenile onset diabetes mellitus. And in case of type 1 diabetes mellitus, there is a complete destruction of beta cells in pancreatic isolates takes place. So it is majority cases are type 1 are autoimmune. And type 2, it is also known as non-insulin dependent diabetes mellitus, or it is also known as maturity onset diabetes mellitus, because it occurs in the middle age or after middle age. Therefore it is known as maturity onset diabetes mellitus. In this type 2 diabetes mellitus, there is no loss or moderate reduction of beta cells takes place, and 90% cases are of type 2 diabetes mellitus. So these are the two major types. Type 1, it is insulin dependent diabetes mellitus, means there is a complete destruction of beta cells of Langerhans of pancreas. And type 2 diabetes, it is a non-insulin dependent diabetes mellitus. So what is insulin? It is a pancreatic hormone. It is a specific anti-diabetic agent, especially for type 1 diabetes, because in type 1 diabetes there is a complete destruction of beta cells takes place. There is no synthesis of insulin at all. Therefore insulin is given in type 1 diabetes patients. So it is a pancreatic hormone. It is a specific anti-diabetic agent, especially for type 1 diabetes mellitus. Insulin was discovered in 1921 by Banting and Best. One is teacher, another is student. It was first obtained in a pure crystalline form in 1926, and the chemical structure was fully worked out in 1956 by Sanger. Insulin, it consists of two polypeptide chains having total 51 amino acids and molecular weight about 6000. And the chain A, it contains two chains. First A chain contains 21 amino acid, while B chain contains 30 amino acid. 21 plus 30, total 51 amino acids. And these two chains are bound together by two disulfide linkages or two disulfide bridges. Here we will discuss structure and biosynthesis of insulin. In body, insulin is synthesized from beta cells of Langerhans isolates in the pancreas. In beta cell, insulin is synthesized from proinsulin. Proinsulin, it is a precursor molecule, or proinsulin consists of three domains. It is a precursor for synthesis of insulin known as proinsulin. Proinsulin consists of three domains: an amino terminal B chain, a carboxy terminal A chain, and a connecting peptide. Means chain A and chain B of insulin molecule are connected by a connecting peptide in the middle known as C-peptide. Connecting peptide is also known as C-peptide. And this proinsulin chain, these three chains are broken by the proteolytic enzyme known as prohormone convertase PC1 and PC2, as well as ixoprotease carboxypeptidase E. These enzymes responsible for breaking of the C chain, that connecting peptide. Here you can see biosynthesis of insulin. Proinsulin molecule, it consists of three chains. By the action of enzyme beta cell peptidase, this proinsulin gets converted into the insulin, and there is a breakdown of the chains, and free C peptide, you can see in the figure, means connecting peptide gets removed, and these A chain and B chains are connected together by a disulfide linkage or a disulfide bridge. Here you can see biosynthesis of insulin. So what is mechanism of action of insulin, how it acts? The mechanism of hypoglycemic action of insulin has proposed that insulin acts by binding with specific receptor on the surface of insulin sensitive tissue. Nearly all the cells or all the tissue having insulin sensitive receptors, such as the skeletal muscle, cardiac muscle, fatty tissue, leukocytes, insulin lowers the sugar content in the blood by turning glucose into the glycogen. Using insulin in diabetes mellitus, it leads to lowering in the level of sugar in the blood and built up glycogen in the tissue. This is the mechanism of action of insulin. Then last and very important point, the classification of antidiabetic agents. So antidiabetic agents, mainly they divide into two parts. One is insulin and its preparation, and another is oral hypoglycemic agent. These are the two parts. In under the oral hypoglycemic agents, there are 5 classes. First major class is insulin and its preparations. Means this insulin nowadays it is obtained by using recombinant DNA technology. First recombinant insulin was prepared by a pharmaceutical industry or biotechnology industry, that is Eli Lilly, in 1982, and that insulin is known as Humulin. Then oral hypoglycemic agents. Oral hypoglycemic agents divided into 5 classes. First class is sulfonylureas, example tolbutamide, chloropropamide, glipizide, glimepiride. Then second class is bigonides, example is metformin and fenformin, but fenformin is withdrawn from market because of side effects. Then next class is thiazolidone diones, examples pioglitazone, rosiglitazone. Then meglitinides, example repaglinide, nateglinide. And last class is glucosidase inhibitors, example acarbose and oglibose.